Spectabilis, dear Blanche, dear members of the board and the scientific advisory board of the Jung Foundation, and dear colleagues. It is such an enormous honor to be standing here to talk about our research, and it's also a very humbling experience given the excellence in medicine here in Hamburg. So I'm really very, very honored. Thank you. The topic that I work on, chronic pain, as you just saw, is actually not a very pleasant topic to talk about, right? Because chronic pain is associated with a variety of disorders, be it direct injury to the nervous system, injury to any part of the body, inflammatory disorders, metabolic disorders, oncological disorders, and so on. And it comes with a lot of suffering. We think, according to the actually very conservative demographic analysis, that every 5th European suffers with chronic pain, and every 10th person in the world will suffer from chronic pain lasting at least 7 years. And every day we are ingesting more than 15 billion analgesic medications. So this could, of course, lead to many a patient to ask the question, would you like a life without any pain? What do you think? Would you like a life without pain? No, because we are scientists and we know that pain has two sides. A life without pain is a horrible life. Most of people with genetically, who are incapable of sensing pain actually have very early deaths and terrible lives, because pain has this Dr. Jekyll, Mr. Hyde character, in that it's both a tyrant in its chronic form as well as an angel in disguise, because it is in its acute form, because it protects us against injury. And indeed, pain is something that had already fascinated Charles Darwin. So in his essay, The Expression of Emotions in Man and Animals, Darwin postulated that physiological pain is essential for the survival of species, and he went as far to call pain an emotion. He said, pain is, like hunger and thirst, a homeostatic emotion. And indeed, this negative emotional aspect of pain, this aversive psychological aspect of pain, actually has an evolutionary importance. It makes sense. Because of that, we are willing to learn to avoid potentially threatening situations. A child learns to sit and to walk because of the percept of pain. But unfortunately, in chronic pain conditions, exactly this emotional aspect of pain takes the upper hand and leads to the most debilitating aspects of chronic pain. And chronic pain patients will tell you that it changes everything. It entirely reformats their life, and they basically also say that they basically cannot tell anymore which part of them is their pain talking or acting versus their own character. And indeed, as the last part of my introduction, I think it's always nice to see how pain has also had a major impact on literature. For example, Virginia Woolf said, let a sufferer try to describe the pain in his head to a doctor, and language at once runs dry. And many artists have also represented pain basically as an expression of their art. And I think the most celebrated and famous one of them is Frida Kahlo, who herself suffered from debilitating neuropathic pain her whole life. So this leads to the question, we have this sort of good pain and bad pain. So why does a physiological protective function get converted into a chronic disease? And can we ever revert this chronic disease? Can we ever get rid of it without also at the same time losing physiological pain? So one aspect that makes pain so hard to treat, chronic pain so hard to treat, is that it is not just a temporal continuum of acute pain. It's not just long-lasting acute pain whereby you can just give acute pain treatment, because it is determined by molecular mechanisms, cellular functions, and also networks that actually are quite different and are not yet well understood. Many years ago, we postulated that although we know basically the textbooks are full of pain pathways, so you could ask, well, everything's known. That's true. There's a lot known about how pain is represented, but exactly which structures are associated with which functions, particularly with respect to specificity for pain and also causality for pain, are not very well understood. we know that over the course of pain chronicity, there is immense remodeling going on in the nervous system, which we call plasticity. But whether this is a cause or rather a secondary consequence of pain is also not clear. And finally, although we know that there are many modulatory systems in the brain, many protective mechanisms, how they are changed over chronicity is not known. So this is not a type of work that a single individual can do. We were very fortunate that the German Research Foundation funded over 12 years a huge consortium with over 40 principal investigators spanning different disciplines, both clinical and preclinical, in Heidelberg and associated universities. So a long time ago, I was very fascinated by this phenomenon of plasticity, and I am aware that plasticity can mean it has very different connotations in different disciplines. In neuroscience, plasticity stands for the ability to learn from previous experience and adapt based on that experience in an activity-dependent manner. And we think that this plasticity could explain the shift that you see here when you look at pain as a response to a particular external stimulus, and you of course have a typical stimulus response function. This function shifts upwards and leftwards in chronic pain conditions, leading to more pain in response to a stimulus, a phenomenon which we call hyperalgesia, and in fact also pain in response to typically non-noxious or non-painful stimuli, which we call as allodynia. And how could plasticity come about? A long time ago, we postulated that plasticity could come about at many different levels, starting from the molecular level, for example, where ion channels may get phosphorylated. This may then lead to change in synaptic function, which can go from a completely silent state to a fully potentiated state. This could lead to changes in cellular responses, such that cells fire more and longer, even when the noxious stimulus has come to an end. And finally, it could lead to network plasticity. One example of it is that the neuronal activity spreads across somatotopic borders, which is a classical aspect of some types of chronic pain. Now, I'd like to give you today a few examples of how we tried to decipher plasticity and why we think that plasticity could be a good mechanism to target for treating pain. Starting with the very, very end or the first part of the nervous system, where physical, chemical, mechanical, what have you, any type of external stimuli meet the nervous system for the first time. This is mediated, for example, by the so-called TRP channels, so transient receptor potential channels. And I'm sure you're aware that Adam Pataputian and David Julius won the Nobel Prize in 2021 for this discovery. Now, these typically respond to different temperatures and different algogens, and very important one amongst them here is TRPV1, which responds to heat but also to diverse algogens. And just to give you now a day-to-day example from our common life, basically every day we feel TRPV1, for example, when you take a very hot shower. But you determine the temperature where you can take this shower, what is the highest temperature that you can take. But what happens when you have an inflammatory injury? You have these inflammatory modulators which sensitize TRPV1 and cause pain, so you cannot accept that heat anymore and you have to downregulate it. Now, it would be very simple if we could simply use drugs against these inflammatory modulators, such as ibuprofen, in chronic pain, but unfortunately it doesn't really work well in chronic pain. And this is because we think that in chronic pain, other modulators come into play. To give you one example, tumor cells secrete, as we know, VEGF, that's vascular endothelial growth factor, endothelial-derived growth factor, which is, as we know, a very important agent that leads to tumor angiogenesis. Now, when we looked at material from patients with tumor pain, we saw that VEGF receptors were not just expressed in blood vessels, as we know, that's the VEGF receptor 2, but also the patient showed an expression of VEGF receptor 1 in noxious or nociceptive nerves, so pain-sensing nerves, expressed a different VEGF receptor, and we observed that this receptor had the capacity upon activation to actually lead to an insertion of TRPV1 in the cell surface, causing more pain. So showing that there are different ways of modulating these channels in a pathological specific manner. Now, two of my colleagues, and I'll just acknowledge my colleagues as I go on, so Dipita Selvaraj and Vijayan Gangadharan then asked, would it be possible to actually target cancer pain? And so we collaborated with the drug company Novartis and obtained an antibody against VEGF receptor 1, which they usually never care about because, that's not the angiogenesis drug. And indeed, we saw that the growth of breast carcinoma metastases in femur bones, and this is very typical in late-stage cancer and leads to absolutely immeasurable pain in these patients, can be reduced. So this is a mouse model here, can be reduced with the anti-VEGF receptor antibody, not by however, the antibody that works on blood vessels. And then as we went on, we found many such examples. For example, we saw Wnt proteins, serpins, semaphorins were secreted by tumor cells or immune cells. And these, as you know, are classically developmental molecules which seem to again be reactivated after injury. So their signaling is reactivated after injury, leading to cell-cell interactions that drive chronic pain. So I showed you now one example of how sensitization of peripheral nerves can lead to increased pain, but plasticity can also be structural instead of only being functional. And if you just look, for example, at the simplified innervation of the skin, we have here pain-sensing nerves, which are naked and superficially ending. And then we have these large diameter nerves. These are our touch-sensing nerves that typically end in end organs like Meissner corpuscles. Now, when we cut these nerves, we see that both types of afferents, of course, regress and are lost. Typically, we then see collateral sprouting from other nerves in this area, and at some point there's functional recovery. But we don't really look at very late stages where these nerves regenerate. And based on some patient reports, we decided to do just that. So we were able to tag in mice, we were able to tag these large diameter tactile nerves and the pain-sensing neurons separately, and then and this is all in false colors. I think this is not working, but doesn't matter. We were able to non-invasively image them in the mouse digit. This is, I would also like to acknowledge that this involved not only excellent work from my colleagues Vijayan Gangadharan and Hongwei Tseng, but also imaging expertise from Thomas Kuner and mathematical expertise from Bjorn Omer, who helped us design machine learning paradigms to be able to test at a very high level of specificity these nerve endings again and again. And uninjured mice show a complete stability of their peripheral nerves. The injured mice, of course, lose their innervation and slowly recover it. And to our great surprise, when in the same mice, so this is the same mouse, and now color-coded to the different types, to the different time points after injury, you see that there's a complete loss of sensation. Slowly sensation recovers, and then it moves in the hypersensitivity range. So these mice become hypersensitive, hyperalgesic, and also allodinic at the time when, surprisingly, nociceptors emerge, but not touch sensing fibers. Now you'd say, why is this surprising? Well, because you're getting pain in response to touch, which should not happen because your nociceptor actually cannot sense touch. So how is this possible? We did electron microscopy and full reconstruction of Meissner corpuscle. So these are the touch organs. And in a normal Meissner corpuscle, you see in green the touch fiber is in touch with these lamellar wrappings that that express the proteins and are responsible for mechanotransduction. But now, upon regeneration and faulty reinnervation of the nerve, we have nociceptors in contact with the lamellar wrappings that actually mediate touch. That means we have now a mechanism where these touch organs are innervated by nociceptors, and of course they signal pain upon touch as a new mechanism for chronic neuropathic pain. Now, unfortunately, most of the drugs that work on the peripheral and central nervous system, there are some types of pain where they are, of course, very effective, but at some point when pain becomes very chronic and is associated with with other changes, that's where you have to look at the central nervous system. And here we know that synapses, of course, can undergo potentiation. For lack of time, I'm not going to go into details, but we and others have described a vicious circle at these synapses between peripheral nerves and spinal neurons, where you have retrograde messengers being generated from the postsynaptic spines that go to the presynaptic neuron and increase neurotransmitter release, again leading to more activation and long-term potentiation. And for some time we thought, well, this could explain why you have instant or very rapid sensitization to pain, but it does not explain the chronicity. So when we dug deeper, we found that several of these molecules actually have the property to migrate, to translocate to the cell nucleus in form of synapse to nucleus messengers that can potentiate pain over days via activating specific gene regulatory programs. And these are the colleagues Manuela Simonetti, Jianing Lu, and Christian New, who did this work, which I cannot show for lack of time, but essentially what they found is that when you repeatedly activate the peripheral nerves, you get neurons in the spinal cord that develop a large density of spines that take up more and more input. Fortunately, you can reverse some of these mechanisms with some pharmacological agents such as cannabinoids. Now we come to a point where the patient in his or her journey has reached a really terrible phase. And this is the phase where, because of pain, they run into stress, for example, at work or at home, that leads to self-doubt, that leads to social isolation, eventually could lead to depression, and new pains can be generated. So indeed, depression and pain are very strong comorbidities. So at some point, you have to look in great details at the central nervous system. And this has been, of course, a major area of study, particularly in human analysis of pain. But we are still uncertain where pain is finally located in the brain. Unlike vision or auditory senses, we do not have one area of the brain that encodes for pain. And work from scientists, particularly with neuroimaging expertise, and we have an absolutely outstanding one here with Christian Büchel in this room, have led to findings where we can start to see some signatures for different areas of the brain that mediate acute pain. There are some areas that actually encode more for this negative, aversive, emotional component of pain that I was talking about. And indeed, we know that pain is highly modulated by expectation and our mental state. And again, there are some important areas that are emerging here. One thing that I have learned as a molecular researcher, I started off as a molecular researcher and became over time more and more a network neuroscience researcher, because I realized that the degree of pain that the human subjects and patients are reporting is actually not linearly proportional to the sensory input. So the phenomena that we look at in terms of sensory experience is not the full story. And pain is, of course, highly context dependent. So the questions that we are addressing now, and where we would like to use the support that is being given by the Jung Foundation, is to try to determine, is there specificity somewhere in the brain for pain perception? I'll give you the example of the cingulate cortex, which is the most highly activated area in chronic pain patients. And however, it's a very large area and could be divided into several subdomains. So here's where we, as mouse researchers, can make a contribution, because by using region-specific optogenetic activation or silencing, we can actually start to determine, this work was done by a postdoc in the lab, Lynette Tan, we can start to decipher which regions are active and important for sensory component of pain versus those that are important for the negative emotional dimension of pain. And then we would be able to, in a patient-specific manner, try to target these regions. And we go, as time goes by, we try to delineate this at a higher and higher and higher cellular level, going to the absolutely precise description of the whole circuitry. Second aspect, which is then therapy dependent, is that these pathways that we delineate could actually also help us improve alternative strategies. So beyond pharmacological drugs and pharmacological therapies, such as neurostimulation analgesia, which is emerging now as a very good alternative, effective alternative in therapy-resistant chronic pain, but is not yet mainstream. And it's not mainstream because there are a lot of studies still needed to determine. There's a lot of variability across clinical studies, and a lot more needs to be done, particularly to understand pathways, which might then help streamline clinical studies since they're so expensive. So here's where, again, we can come with our mouse models and try to reverse translate neurostimulation analgesia in animal models and try to improve clinical efficacy. And because the motor cortex is the area that is most highly seems to be most highly associated with neurostimulation analgesia, which is completely, absolutely not something you would expect, because after all, the motor cortex is supposed to mediate motor functions, how we move and so on. And there was a lot of skepticism whether it is really the motor cortex that is leading to the analgesia. We said, well, that's something we can address in mice. So we went in the motor cortex of mice and used an alternative approach, chemogenetic activation, where we specifically could activate the motor cortex. And we saw that led to a strong reduction of neuropathic pain, not only of the sensory component of neuropathic pain, but also of the negative affective dimension of pain. And we do this kind of work using operant chamber experiments, where we let the mice decide and avoid or prefer certain areas where they either get pain relief or they feel more pain. And indeed, the mice showed that they preferred an area where their motor cortex was activated in a highly specific manner. And then, using methodologies that are the advantage that mouse models can offer, we could then come in and delineate the exact pathways. So what our findings led to is that we can now say that you actually need to activate the deeper areas of the motor cortex in order to get more analgesia. And here are pathways. There's a dichotomy of pathways that mediate the sensory or modulate the sensory or affective dimensions of pain. And so this can have clinical implications. For example, one may need to go deeper with more or better transcranial magnetic stimulation regimens, which are becoming available. And secondly, one can use the activity of some of these areas as biomarkers to test for efficacy of regimens, which can then be rolled out in patients. And this work was done by Zhengang and an anesthesiologist in the lab. As my final aspect will be to try to now make the big gap to psychology, because this is where psychological and psychiatric mental disorders are something that go hand in hand with pain. Yet in our field, although biological factors are being studied, that would be me, for example, others are studying social factors. Some people do. Some people focus on psychological aspects. We typically work in our own silos, and this is what I believe is also inhibiting or hindering progress. So we see a need now to intercalate our research, our mechanistic molecular research, with psychological and psychiatric aspects. And we started doing that with the example of fear, because fear is something which is very, very, very rampant in our world. Fear is important for survival, but fear can also be debilitating. And in a way, you can consider pain and fear as two sides of the same coin. Ironically, acute fear suppresses pain because it enables you to escape and so on. But in psychophysical studies, chronic fear, when we chronically have a fear syndrome, we tend, and if we have anxiety traits, we tend to exacerbate our pain. And this could be a mechanism for exacerbation of chronic pain. So the question here we ask is, what is the neurobiological mechanism? Because actually the brain areas that are activated during pain and activated during fear share many commonalities. And as the last slide here for data, I'd like to show you how we can then still use a molecular approach by which we can selectively tag neurons that are activated, for example, during pain or during fear, and we can identify these neurons. So, for example, in red, we have identified neurons in the prefrontal cortex that are activated during fear and mediate fear, whereas the green ones are the ones that are activated during pain. Now, you see, these are actually two separate populations, typically, with a small overlap. But surprisingly, when you have a chronic pain state, you see that these populations start to overlap. That means that if I have chronic pain and I'm feeling pain, at the same time, I'm triggering fear responses in my body, and vice versa. When I feel fear, I could be exacerbating my pain, because it also opens up now the pain inputs to a variety of brain regions that are actually associated with negative emotions. So the corollary of that would be, then if I were to inhibit neurons that mediate fear, so if I inhibit the fear recall neurons, how would pain be affected? And this is an experiment you can do in mice. And when we did that, we saw that we actually had a significant reduction in neuropathic hypersensitivity. And this work was done by Lena Stegmann, Shenglu, and Oskar Retana. So we postulate, and this is the main project that I would now like to going ahead do with funds from the Jung Foundation. We postulate that exposure to pain leads to induction of fear. And then when you have a second hit in form of some sort of tissue injury or an inflammatory condition, that would lead to an exaggeration of pain. And then the corollary of that would be, in that case, if that's true, then we should be able to treat chronic pain by extinguishing long-term fear. Indeed, there are approaches in cognitive behavioral therapies that attempt that, but we would like to try to improve that by bringing in pharmacology and neurostimulation. So I'll end with this slide. We totally believe in multimodal therapy for chronic pain. Each type of therapy, physiotherapy, cognitive behavioral therapy, neurostimulation, and of course, a mechanistically targeted pharmacological therapy are important in their own right, and they need to be combined, they need to be matched. But we are going beyond this era of multimodal therapy to the era of personalized therapy, and this is where I hope that we preclinical researchers can also make a contribution. So at the end, I'd like to thank my absolutely colorful research group for their immense efforts, and our grant-giving organizations. I am extremely thankful to the Jung Foundation, because the flexibility of the funds actually is very empowering in terms of helping research careers of young scientists. And this is something that I think this tool, therefore, has very long-reaching consequences that will go beyond our work. In the end, I would also like to thank my university with its motto, Semper Apertus, always open. And I thank you for staying so late and paying attention to my words. Thank you so much.